For today's session, we will answer both papers of Feb-March 2024. These are the recent exams, so it's a great opportunity to work on both papers to check the style of the questions. Both exams were extremely easy, has different ideas, a lot of ideas, but both were extremely easy. And I want you just to focus on everything in paper 6 and the topics included in paper 4 in this paper, and you should know how to write the answer accurately in this paper. We will answer both papers in the time of the session. Don't worry. We can definitely answer both papers in the time of the session. Let's start directly with the paper 6. Paper 6, as usual, two questions. The first question is the experiment question, and the second question is the question of the magnification, the calculation, and the food test, all of this part, and plan and investigation question. Let me check with you the question. As I told you several times, يا ولاد في class, please, whenever you have an experiment, you should read the experiment first, then start answering everything. Because now, Yadan, while reading the question, you will realize that there are some points he might ask you about, and I will show you how. A student investigated the effect of glucose concentration on the rate of anaerobic respiration in yeast. So we will check the concentration. If the concentration is different, how this reflect on the rate of anaerobic respiration. Wow, nice that Malak joined, because I know that she might got panicked of paper 6. Hi, Malak. We just start answering this question, Feb-March 2024, and we start reading the experiment question. Anaerobic respiration in yeast breaks down glucose to form ethanol and carbon dioxide. Anaerobic respiration in yeast causes the blue dye methylene blue to become colorless, and the time taken for the blue color to disappear can be used as a measure of the rate of anaerobic respiration in yeast. So he's telling you now that the time taken, the idea of the time taken is the dependent variable. That's what I'm saying, Malak. You could extract the information needed for some questions or for the answer of some questions even before reading the question. مفيش question experiment, he will not ask you about the dependent, independent, and control. So now we know from the first part of the experiment the dependent variable. The student used this method. Label one test tube 0.0%, one test tube 0.5, and another test tube 1%. He put 5 cubic centimeter of water into test tube labeled 0%, and I can definitely now expect, this test tube is a control one because it has no glucose concentration, Ya Abdurrahman. No glucose concentration, so we consider this as a control part of, or control experiment, or control part of the test tubes offered. For step 3, he put 2.5 cubic centimeter of 1% glucose, 2.5 cubic centimeter of water in the test tube labeled 0.5%. He put 5 cubic centimeter of 1% glucose solution into test tube labeled 1%. So he changed what, Yamalak? He changed the concentration of glucose. So now we know the independent, which is the concentration of glucose, and the dependent, which is the time taken to change the color of methylene blue from blue to colorless. In step 5, he stirred the contents of the beaker containing the yeast suspension with the glass rod. He might ask you about the reason of stirring, and definitely this is to provide uniform distribution of the yeast in the solution. So now we might find a question like this. Add 5 cubic centimeter of the yeast suspension. Now we have a control because he used the same volume of yeast suspension in all tested tubes. So Yerim, now you realize that we have a control here. He put the all three tested tubes into a water bath at 40 degrees Celsius. Another control, Yedan. The temperature was 40 degrees Celsius in all tested tubes. He start the stopwatch and wait for 3 minutes. 3 minutes waiting for the effect of thermostatic water bath or the water bath that he used in order to distribute again temperature evenly for the 3 tested tubes. After 3 minutes, he removed the tested tubes from water bath and he placed them into a rack. He added 1 drop of methylene blue dye. This could be a method of improvement, Yarim. Why? Because he used 1 drop. الدروب دي ممكن تكون like this, الدروب like this, drop like this. So we could use this as a method of improvement. We should detect the main volume of methylene blue instead of saying 1 drop. But this is our expectations. We will check the question. Use a second pipette to slowly add a layer of oil to each of the tested tube. And he gave you now, Yahya, the thickness of the oil layer. It is 1 centimeter, and this is another control. If he asked you up to 3 control variables we used in this experiment, we can give him an accurate answer. If he didn't write, Maya, that the layer of oil to maintain anaerobic conditions, I would expect this could be a question, in importance of the oil layer. And definitely it will be an easy question, it's okay. This to prevent the oxygen and to provide the anaerobic conditions or absence of oxygen in the reaction. Okay, there is a question from Dan. How is the layer of oil is a control measurement? Because he is giving you the thickness of the layer, Yadan. Okay, so it is 1 centimeter for all tested tubes. So you will not write the oil drop is the control. You will write the thickness of the oil. It's totally clear in front of you, and for all of them, it was 1 centimeter for the tested tubes contents that we have. You got it? Question step 12. Put the tested tubes back into water bath and restart the stop clock. And he measured the time taken for the blue color in each of the tested tubes to disappear, and he wants you to record the time taken in seconds for each tested tube. And he's giving you the time, but honestly, يا براء, the time was in minutes. So we need to convert it to seconds. And he told you, Irene, that for 10 or more than 10, you can record the results as more than, I don't know why it's not writing, more than, what's happening? Yeah, 600. So we will convert the time into seconds and we will draw the table. Okay, could you please help me to convert the time into seconds? I think this one is easy. This one is 132 because 2 times 60, 120 plus 12, 132. For the 6 and 2 minutes, 6 times 60, 360. So this is 362. This is the time in seconds here. And about 10 times 60, it could be 1,000 or more than 600 or more than 600. And he wants you to draw the table. As I told you several times, how could we draw a table, يا يحيى? Using a pencil. طبعا اللي أنا بعمله ده not that accurate. We should use a pencil. divide it into two sections or two rows, two columns. The first one, you should write the heading here, and this is the independent part. And always, this is the independent part. Independent part comes first. So the independent here was what we are changing, and it is the concentration of glucose per percent. And we will write here 0. And the second one was 0.5%. And please don't write percent because you wrote it in the heading of the table. And the last one is 1. And here we will write the time needed for color change. It should be per seconds because he asked Juyerim to write the time in seconds. Okay, so the first one for الزيرو it was more than 600 وهو اللي قال لنا كده يا عبد الرحمن. هو he mentioned انه for 0 it should be more than 600. For 0.5 it was 362, and for 1 it was 132. And now we got 4 out of 4 in our table. But please don't forget ان انت you would use pencil in the drawing of the table, and please don't forget انه headings should be per unit. اوعى تكتب headings without unit وحط data. وبليز 1 mark will be awarded for the conversion of minutes to seconds. Is there any problem in the table? Is this clear? Let's go. If you have any question, please text me the chat box or turn your mic on, ask your question directly. Yahya, how are you now? Are you better?
Alhamdulillah.
Alhamdulillah, ya Habib. Alhamdulillah. Okay. Based on the results provided, what is the conclusion of this experiment? What is the conclusion of this experiment? As concentration increase, what happened for the rate of respiration? Okay, what happened for the rate of respiration? The conclusion, not a reading from the data يا ولاد. Conclusion, as I told you several times in Arabic. من الآخر الexperiment دي بتقول لي إيه؟ That's what I want you to write, and it is 1 mark question. So you should write the greater. No, ya Abdurrahman, please don't link it to the time because he's not asking you to describe. Describe is paper 6 question. You need to write something about the main objective of the experiment. So we will write a greater rate of respiration at a higher glucose concentration. How بقى يا Mr. Abdurrahman will ask me now, طب ازاي يعني ازاي? That's what you concluded from the results, but we will not write about the results. You got it, Yahya? I will not write about the results because this is a conclusion. The conclusion related to the objective of the experiment. The student Yamalak was investigated the effect of concentration of glucose on the rate of respiration. So you should link your conclusion to the objective of the experiment. Okay, and this is the conclusion. State the independent. Yeah, we said the independent several times, the concentration of the glucose. If you want to say any other answers, please let me know. It will be better if you turn your mic on and ask, but if you don't want to, please write in the chat box and I will check between now and then. State 1 variable that was kept constant. We said around 6 Yamalak variables kept constant. Like we mentioned, the concentration of yeast, or volume of yeast, he used 5 cubic centimeter. من اليست. We mentioned the total volume of the glucose and water. This was fine. And we mentioned the type of yeast that we used. It should be a control. The time for the equilibrium, 3 minutes for the equilibrium, or 3 minutes for the even distribution of the yeast with temperature, it was a control. The temperature, 40 degrees Celsius, was a control. The volume or the concentration of the methylene blue considered as a control, as he used a pipette, but we could improve this. So all of these are control. We mentioned like 6 controls, and I want you to write one. That's why it is an easy question. I will write temperature, and I will go with you to the next question. For question V, explain why it was important to stir the yeast suspension in step 5. We expected this question, and we said the idea of the even distribution or uniform distribution of yeast fill solution. So this is the answer. Question BI. One way to improve the investigation would be to use an increased number of different concentrations of glucose. He wants 2 other ways to improve the investigation. أنا دايما بقول إن إحنا لما نقلل الانترفلز this will be a method. طبعا I want 2 other ways to improve this investigation. What do you think? هل هو يا ريم mentioned anything about the repeat of the experiment? He did not. When we read the experiment together, guys, he did not mention that he repeated the experiment to calculate the mean or the average. So it could be a method of improvement. So you can write repeat the experiment, okay? And this will give you repeat at each concentration. This is a way, okay? This is a way. Yes, ya Abdurrahman, the idea of the drop of methylene blue, we should use a fixed volume, okay? Or volume, we could get it and make it standardized. Is it easy to detect the color change without calorimeter؟ أنهي الأحسن يا عبد الرحمن؟ إن أنا decide إمتى الcolor change؟ ماجد will decide, عبد الرحمن will decide, Dan will decide, Sahar will decide. It could be a human error, but if I use the calorimeter, this will be perfectly fine. This is a device we will use in the lab, and now we have digital calorimeter, so you will put just a color, fix it, and measure. خلاص, this will be a method of improving the measurement. Okay. We can do different concentration at the same time. This is another way of improvement. So I mentioned with you now 4, guys. You can pick any 2 to get the 2 marks of this question. Describe how you would make 5 cubic centimeter of 0.25% glucose solution using 0.50 glucose. Yes, salam. I have 0.50 glucose, and I want to change the concentration into half, to drop the concentration into half. Okay, and I'm using 5 cubic centimeter. يبقى simply I will use 2.5 من ال 0.50% glucose and 2.5 distilled water. وموست probably يا ملك لما يجيب لي question like this, the readings will be direct. The readings will be the same. Not the same, I mean close to each other. I mean direct, يعني مش you will not exert effort in calculation. دايما I mean the same ايه، دايما بيconvert من الhalf لل 25% او من 50% ل 25%، و I think it's easy. There is a formula to do this بس هنعرفها في ال AS biology. For the time being in the O level, ال 5 cubic centimeter دي I will take منها 2.5 مش عارف ليه البن بيعمل كده بس 2.5 cubic centimeter from ال 0.50% glucose و 2.5 من ال cubic centimeter distilled water. And now we got the 2 marks. Let's go to triple I. Describe the method you would use to test a solution for the presence of glucose. Usually this test is a 3 marks test. Do you know what is the test for reducing sugar for glucose? The name of the test? And I gave you a hint now. Usually this question coming in the exam as 3 marks question. But thanks God in this exam it was 2 marks, so we'll write 2 points. What is the name of the reagent? Benedict. Exactly, Benedict. So we will just write, we will use Benedict and we will heat the solution. And there is no need to talk about the results because he's asking about how you would use a method to test the presence of glucose, and he's not talking about the results here. I will write literally 1 sentence. I will add Benedict solution, then heat for 3 to 5 minutes. Okay, let's go to question C. A student
Mr. Magid?
Yes, Yamala.
Mr., can you go back to the previous question? Because my internet was lagging for...
Okay, this one?
Yes, this one. Yes, can you just explain it?
Yeah, no problem. Okay, he's giving you the final volume that we are using, or he asked you to prepare. He asked you to prepare 5 cubic centimeter of 0.25% glucose, and all what you have is 0.50% glucose. So you need to drop 50% or 0.50% into half, into 0.25. So we will use 2.5 cubic centimeter of the 0.50%, add the other half of the solution with distilled water. Add distilled water as the other half of the solution. And I told them, oh, Malak, if your internet was lagging or was not that stable يعني إن usually الفوليوم اللي بيask you to prepare والconcentration اللي بيask you to use بيكونوا numbers you could or values you could use without calculation like ال 50% وال 25%, something like this. Because he wants to check, how could you make a dilution technique or a dilution method in that way or not. Okay?
Okay,
is it
thank
easy
you.
for you? You got it?
Yes, thank you.
Okay, great. أنا عارف paper 6 is your favorite. But so far it's easy. So far it's an easy paper, yeah. Okay? Type. Now, a student investigated the effect of temperature on the rate of respiration in yeast at 25 degrees Celsius and 35 degrees Celsius. In one experiment, the student measured the volume of carbon dioxide produced by the yeast every 5 minutes for a total of 30 minutes. I'm not reading that fast, ya Yahya. I want you to read using same pace in order to save time and to give you time to think in the exam. Please try to practice on this way. Describe suitable apparatus that you could use to collect and measure the volume of carbon dioxide produced. ازاي بجمع يا يحيى الجاز any gas by using gas what? Collect gas from an experiment by using gas what? Gas syringe. Okay, this is the apparatus in the lab, one of the lab tool to collect or measure the volume of carbon dioxide. Okay, so using gas syringe. There is another method. I think, Khalaf, you need to mute your mic. Thank you. There is another method, and I think you took it in chemistry, guys. Okay. The measuring inverted, no problem يا حبيبي. Measuring inverted or the inverted measuring cylinder with a delivery tube. Okay. I think yeah, Dan, you took it in chemistry, right? The measuring cylinder اللي هي بتكون upturned measuring، انا بفتكر الكلمه upturned measuring cylinder او inverted measuring cylinder with delivery tube. بس it's way easier in biology to write gas syringe. Would go to the next question. Using a gas syringe, this will be the method of collecting carbon dioxide. The first question, Yadan, in this exam, I will ask you to answer without checking the data or the question. What's meant by anomalous results? One line definition: results does not fit what? The trend or the pattern. Okay? One sentence, ya Malak ya Ahmed, one sentence. Any result in the experiment does not fit the pattern is an anomalous result. Okay, my question now, ya Malak Zakaria, what is the anomalous result here? Experiment 1 ولا 2 ولا 3?
2.
Experiment 2, bravo. Experiment 2. Experiment 1 is 1.8, experiment 3 is 2. So only experiment 2 has value out of the trend or out of the pattern. So experiment 2 هي anomalous results. Let's go to question triple I. Describe how the student calculated the mean volume of carbon dioxide. He's not asking you to calculate. He's asking you, ya Yahya, to clarify how the student calculated this. تفتكر ازاي؟ How could we calculate the mean here? عملناها ازاي؟ We should add the value of what experiment? Experiment what and what? Experiment 1 and experiment 3. So we should get experiment 1 and experiment 3 divided by total number. Okay, so we should include, or we should use in calculation, experiment 1 and 3 only. There is no need to talk about the calculation because it is 1 mark. So you need, all what I want you to write is how could you calculate experiment 1 and experiment 3 only in this experiment. Okay, another question. He's giving you the results. The time was 5, 10, 15, 20, every 5 minutes interval. He measured the volume of carbon dioxide at 25 and at 35. And he's asking you for 2 questions to compare the mean volumes of carbon dioxide produced at 25 and 35. What should be your answer? ازاي compare؟ وخدوا بالكم compare is not a description. I want you to compare between the 35 Celsius degree results and the 25 Celsius degree results. How could we write a comparison? Overall, Yareem, carbon dioxide produced at 35 is more than the carbon dioxide produced at 25 or not? And this is a comparison point or not? Definitely yes. So we can say in overall, the amount of carbon dioxide produced at 35 degrees Celsius per cubic centimeter is more than 25 degree. This is a point. What else? Yes, يا ملك.
مستر ممكن أقول إن في 25 و 30 minutes عند 35 temperature هما كانوا constant.
Yes, you should write levels of or constant or the same. This is another point. However, at 5 and 10 minutes, it was no carbon dioxide produced at the beginning of the experiment. So we could mention this one, okay? And we could mention this value because this is a comparison. And هتقولي على فكرة في 25 and 30 minutes, carbon dioxide produced was the same. And at 25 degrees Celsius, at 5 and 10 minutes, it was 0. Okay. And خلاص 2 marks. And what we mentioned give you 3 marks, not only 2. He wants you to plot a line graph on the grid of mean volume of carbon dioxide produced against time using all of the data in table 1.2. You will need to plot the data for each temperature as separate lines on your graph include a suitable key. Fine. أهم حاجة عندي إن هو he wants 2 line graphs for 25 and 35, and the time is the independent variable. So I should put the key. Please don't forget this. For example, I will use X for 25 degrees Celsius to plot the point of 25 degrees Celsius, and I will use the dot for all of the points of 30 degrees Celsius. Regarding axis, for the x-axis, we will write time per minute, and for the y-axis, we will write the volume, and we will just copy this from the table. The volume of carbon dioxide gas per cubic centimeter, and التايم easy جدا. It will be 5 10 15 20 25 30. And برضو يا ولاد imagine الحمد لله, thanks God for this exam, intervals will be nice because you could use from 0 to 3.2 even per point. Okay, so we can definitely say, for example, if this is 1, this is 2, and this is 3, we already passed more than 50% of the grid, and we can assign like 2 or 4, I think 4. Yeah, 4, 4, 4, 4, and 4. أنا معلش مش شايف قوي يعني في الجريد، بس we can definitely assign إنه دي 0.2 0.4 0.6 0.8 وال1. نقدر نقسمهم كده عشان ما نكتبش كل حاجة. و you can definitely pick الvalue اللي already added أو inserted in the table. And you will draw the lines, line for example, 25, another line for data of 30 degrees Celsius. You should connect between the lines using a ruler, or connect between the points. If it is curved, draw the line graph. And he's asking you to estimate the time taken to produce 3 cubic centimeters. So handle 3 exactly, and you should draw this, please يا عبد الرحمن. Don't do it كده by sight. لازم لأنه عليها mark إن أنا I have to show him on my grid how could we get the estimated number. Okay. He said L3 بنتشيك time taken, and you will write the time per minute here. It is 2 marks question because you will show him on the graph how could you get the data or the minutes. Okay, let's go to the last question in or the D question on this question 1. Carbon dioxide gas was bubbled through hydrogen carbonate indicator solution. The indicator was red before the gas was bubbled through, and he wants you to state the color change that would occur. The color change will be from red to what? Because carbon dioxide is an acidic oxide. In the acidic conditions, the color should be yellow or orange. Both are okay. So this is the change of the color. It will be yellow or orange. Now we got 27 marks of the first question. Let's go to the second question quickly. In the second question, he is asking you, he gave you a red onion, the cells. It was really easy question, and he's asking you to calculate, to draw a large diagram of the cell labeled X in figure 2.1. Okay, so we will draw the cell, and it's easy. I will draw it here عشان تشوف الcells. So I will draw the cell like this, which should be that large. وأنا معايا ruler, which it should be all unbroken line. And don't sketch, please. I will draw inside the cell membrane. I will draw the nucleus in. And if you did not shade, no problem. Okay, you will draw just a circle, represent the nucleus. It's okay. And if I will be in your place, I can see like a 3D, or I can say 2D shape of the cell wall. So I will draw another line here. معلش, I cannot control it well عشان الديجيتال pen ده. بس it should be thinner than this. Okay. وكده أنا هاخد الفول مارك في الدروينج يا ولاد. Okay. This is cell X, and he's asking you for the next question to measure PQ, and you should measure it in millimeter, and the magnification is here, asking you to calculate the actual length. How could we calculate the actual? Using IAM formula, image size divided by magnification, and the units provided is in millimeter. So I will not convert the units ولا حاجة. I will measure in millimeter, substitute, get the reading, but the most important thing that your answer should be in 3 significant figures. Please don't forget to read the question to the end. Another question. Last question in this paper. Imagine we finished this paper in almost 40 minutes. We can say 45 minutes because we might take 5 minutes in writing this plan and investigation. Okay. Water moves into and out of cells by osmosis. Plan an investigation to determine the effect of the concentration of sodium chloride solution on osmosis in plant tissue. I want you to think with me, how could we write this plan and investigation point? I told you several times, point for the setup, dependent, independent control, procedure and method of measurement. Finally, you will repeat the experiment for reliability, and safety precautions should be the last point. If you followed this, you will get the 6 marks in this question easily. Okay, so any one of you can turn the mic on, trying to tell me how could we carry out this experiment? Anyone, يا ولاد. And no problem at all if you don't know the some of the steps. I will help you to think with you about this. No one? نايمين ولا ايه؟
بس ممكن أحط ال plants في different concentrations.
So this is your setup. Okay, I will prepare. طيب خليني اقول لكم simply كده يا ولاد قبل ما نكتب. عشان اعمل الاكسبيرمنت دي, what do I need? I need beakers, for examples. Okay, for example, I need 5 beakers. Okay, and one of them will be distilled water, and then I will have different concentration. I can say 0.50 concentration of sodium chloride. Sodium chloride, the table salt يا ولاد. Okay, I can say 1, I can say 2, and I can say 5%. Okay? And I will put part of the tissue of the plant, where I should mention in a plant is the same type of plant or same species of plant, same age of plant, same area we got the tissue from. All of this are right.
بعد كده أقول independent والdependent.
أنا لسه ما اتكلمتش على الرايتنج يا ملك، خلينا نكلاريفاي الكويستشن وبعدين هنكتب بقى مع بعض. Okay? Then you will measure what? I can measure here the difference in mass from the beginning of the experiment and the end of the experiment. بس عشان أعمل كده يا ملك I have to mention إنه I will measure the mass of the plant tissue at the beginning of the experiment, and I will measure it by the end of the experiment to get the difference. From this experiment, Yadan, some plant cell will take water and some plant cell might lose water. So the measurement will be based on the difference in mass. You got the idea of how could we measure? Repeat. Safety precautions, as long as we have any salt, even sodium chloride, we should wear goggles, wear gloves. I think now if I ask same question, you could write the answer with me. وتفضلي يا ملك قولي لي بقى هتكتبي البلان بتاعتك ازاي.
هقول أنا هجيب البيكرز وأحط فيها different concentrations
اوكي.
وأحط فيها هو الكونستنت هيبقى اللي هو same volume of concentrations و same volume of distilled water و same plant.
برافو عليكي. same volume و same plant. We could say same temperature, by the way. We could add this in our method and we will do this at same temperature by using thermostatic water bath.
Independent اللي هو effect
What
of
we change the concentration of sodium chloride.
والdependent اللي هو effect of the concentration على الماس of the plant.
لا هكتبها ازاي؟ It should be something measurable يا ملك. It should be something measurable. ايوه exactly. الماس difference between starting والending mass. The difference in mass could be in dependent variable. And you mentioned the control. هنقول ان هنسيب البلانت for we can say any valid amount of time. We can say 30 minutes, we can say 20 minutes, we can say 10 minutes. أي يعني reasonable amount of time. هنتشيك الماس after التايم assigned period of time. وهنrepeat experiment for each concentration to calculate the mean, and we will add a point of safety precaution. Now we have covered this paper 6 ويعني أتمنى إن الإكزام بتاعك يكون like this. It is an extremely easy paper. Do you have any problem in this paper يا ولاد so far? حاسين بأي مشكلة ولا it's exactly like what we are answering in the class? I think nothing different, nothing new. Easy paper, and we learned more how to get the full mark in this paper inshallah يعني hopefully. It's easy to get a full mark in paper 6, by the way. Let's go to paper 4. Enzymes are catalysts. Describe what's meant by the term catalyst. It is 2 marks. Okay. So, Abdurrahman, what's meant by term catalyst? If you want to write it in the chat box, fine. If you want to say it loudly, it's okay. طيب ملك عماد.
It's a substance that like increases the rate of the reaction without being chemically changed
changed.
by the reaction.
Yeah, without being changed. So chemical substance change the rate of reaction without being changed. We got the mark. Okay. Yes, without being affected. Okay, Abdurrahman. Enzymes are proteins. State the names of the chemical elements found in all protein. So Dan, what are the chemical elements forming protein?
Nitrogen, carbon, hydrogen, and
And
oxygen.
oxygen. Okay, I think يعني question اخد عليه 2 marks ده كادو. Okay, another question. Graph, and I'm asking you graph about the pH and the enzyme activity or the percentage activity. And I'm asking you to describe and explain the change, and it is 6 marks. We have a question like this in our classified, guys, if you remember. Exactly like this. And I told you 3 marks for description and 3 marks for explanation. I will not write with you the answer just to save time. بس خلينا نcheck how could we describe this graph. Reem, can you describe this graph for us or the results that we have? Are you able to talk,
You
Reem?
can say no, as pH increases.
Yeah,
Yes.
okay, okay.
We can say no, as pH increases, activity would increase and then decrease.
دكتورتي دي اتعلمت مني ايه how to steal اللي انا بقولها كل مرة يا دان قولي لي how to steal the first mark from examiner. She mentioned أول حاجة إنه what increased, and then she will definitely say something about the optimum and what happened after. If you want to link a description to the explanation, fine. If you want to describe first, then explain, it's perfectly fine for me. I'm sorry for interruption يا ريم, هسيبك خالص لحد ما تخلصي يلا.
And most enzymatic activity was at pH 7, so it's like the optimum temperature, the optimum pH.
Yeah, optimum pH, yeah.
And then there was no activity at pH like 4 or below 4, and then 10, more than 10.
Okay, so you need just, this is called what, Reem? This is called comparative. No, this is described, but this is called comparative manipulation. يعني إيه؟ يعني أنا compare using data, which is extremely important يا ولاد. بلاش compare without taking any data. What she mentioned is إنه ال pH 7 is the optimum at which the percentage concentration or percentage activity was 100. However, she can talk about activity is 0 or at low and at extremely high pH. يعني هي كانت قدرت تقول إنه at almost 4 or before, يعني ممكن نقول 3.8. okay, 3.8 وهنا we can say, and I'm already I covered the point. We can say at 10.2, okay, it was 0. This is a description. So I think we need to move to the explanation. Bara, could you explain this graph? You need to talk about بقى الactive site والsubstrate والpart اللي احنا already حافظينه ده. Could you please clarify?
Yes.
Yeah, I know that you can do it. ايه الفيرست بوينت اللي هتقوليها وان انت في الاكسبلانيشن وبرضه هتسرقي بيها مارك من الاكزامينر؟ ان اللي تشينج في البي اتش affects the shape of the lipase or not, affects the shape of the enzyme or not. خلاص بقى انت مش هتكلميني per data, هتكلميني بس about your biological background. يلا كملي خلاص, انا مش هقاطعك تاني, سوري. كملي بقى اللي عايزه
Okay, so the substrate find its active site to bind with it and form the complementary active sites.
شطارة
found
شطارة. شطارة. برافو.
And
well done.
a form enzyme substrate complex, and when it reaches the optimum pH, the enzyme start to denature, and the active site, the substrate, will no longer fit it to its active site as it changes the shape.
Exactly. ما شاء الله. Perfect. So Reem وبراء together will get 6 marks about the description and explanation. Let's go to CI. Complete the word equation to show the substrate and product for lipase. يحيى, a lipase enzyme break down the lipids into what? Can you remember the product? You're not in the mood يا يحيى؟ لسه تعبان ولا ايه؟ ولا انت مش في الميتنج ولا ايه؟
مستر أنا مش سامع أي حاجة.
ليه يا يحيى مش سامع؟ ودنك اللي تعباك ولا الساوند اللي فيه مشكلة؟
مستر ممكن أقول أنا؟
يحيى I'm asking literally. انت مش سامع عشان انت تعبان ولا انت عندك لاجنج ولا ايه؟
أجاوب يعني.
انا عايزك تفكر معايا في I. الليبيدز will be converted into what? ايه البيلدينج يونتس بتاعت الليبيدز when the lipase break it down? Fatty acids and what?
جليسرول.
ايوه يا بابا، مش اللايبيز ده اللي بيبريك داون الليبيدز into fatty acids and glycerol. اوكي مايا. double I. Lipase acts in the duodenum. Explain how the body provides a suitable pH for lipase activity. What do you think? Is food coming from the stomach in an acidic pH. ازاي بقى البادي provide suitable pH for lipase activity in the duodenum? By what? Try كده to remember. ايه الستراكشر اللي will be produced by a liver to change the pH. I'm giving you a hint now اهو. اللي بنستورد في الجول بلادر. Does anyone know the answer يا ولاد؟ البايل. Exactly. Who's answering? خلف ولا عبد الرحمن ولا مين؟ Who said البايل؟ عبد الرحمن. ماشي يا حبيبي، مش سامعك قوي. Okay. So for البايل you will take the first mark, but you need to talk about what we mentioned. إن هي produced by the liver, released from the gallbladder to provide alkaline pH, alkaline mixture. With this will neutralize the gastric juice or الفود اللي coming from the stomach. طبعا what we mentioned يا عبد الرحمن is يعني might be more than 3 marks بس. نايس. عرفتيها دلوقتي او افتكرتيها يا مايا؟ البايل والميجر رول بتاع البايل بيعمل حاجتين يا مايا. نمبر 1 بينيوترلايز الفود اللي coming from the stomach in the duodenum. ونمبر 2 بيعمل emulsification. Emulsification, it means if we have large globules of lipids, it will break it down physically into small droplets. Maya, you got it?
أيوة مستر ثانك يو.
اوكي انا عارف ان انت عارفاها، بس يمكن يعني نسيتيها بس، لكن انا عارف ان انت عارفه البايل والفانكشن بتاعتها. اوكي let's go to question 2. تهيألي question 1 ده كان كادو يا ملك يا عماد. اوكي يلا question 2. خلف, could you please answer with me question 2? I will do AI. I want you to think about double I, please. For AI, I want you to label with an R the position of the right ventricle. This is the right side. This side is the right side, and this is the ventricle, so I will put R here. Okay. Next question. Hey Khalaf. State the names of the structures labeled A and B on figure 2.1. What's A and B? Khalaf, can you hear me? Are you in the meeting? You are in the meeting. Safe, you are in. Would you please answer with
Yes,
me,
sir.
Safe? يلا. أو Khalaf, any one of you, if you know the answer. يلا. What's A and what's B? I'm asking about the valves. What is the name
Yeah.
of the valve A?
Bicuspid valve.
Yes, exactly. Bicuspid valve, or you can say atrioventricular valve, as it is a separator between atrium and ventricle. And what about B?
I don't know, but I'm pretty sure tricuspid
No,
valve.
the
actually, I don't know B.
طيب fine. The
Semi-unit.
بص يا سيف, B or same in the same location on the left side called semilunar valve. Semilunar valve. Okay, so B is the semilunar valve, and
the tricuspid valve has
yeah,
three.
this is the tricuspid valve.
It has
اللي هي
three.
المرسيدس sign, as we said
Yeah.
before several times. Okay, so A is the atrioventricular valve or bicuspid valve, and B is the semilunar valve. You will take the 2 marks, and that's it. Question triple I. Complete the sentence about blood vessel C. Blood vessel C transport blood from what to the what? I'm talking about blood vessel C. Okay, from what to the what? From the left ventricle. Oh, please don't say from the heart. From the left ventricle to what, Yadan? From the left ventricle to what?
Sorry, sir, I couldn't hear you well. Can you repeat it?
Yeah, I'm asking about blood vessel C. It takes blood from the left ventricle to what?
Aorta.
This is the name of the aorta, of C. This is the name of the blood vessel. الأورتا بقى بتتيك البلاد to فين بقى؟
The body.
Yes, exactly. To the rest of the body. I don't want any other answer. To the rest of the body, you got the second mark. Okay?
Okay.
Yeah. Let's go to B. Malak Ahmed. This is a cross-section of part of a heart that has an incomplete structure. And the incomplete structure is the separator between the atria and the atria, or the right atrium and the left atrium. I mean, the atria, this is the plural of the atrium. There is no atriums word, okay? We called it atria. Explain how the incomplete heart structure may affect a person's ability to transport oxygen, and it is 3 marks question. How could Yamalak steal the mark of the from the examiner, the first mark? You should say this is a hole in what structure? What is the name of the separator, the barrier?
The septum.
Exactly, the septum. So
The
we
septum.
can say this hole in septum, so left atrium joined to the right atrium. What will happen after is مين any type of blood will be mixed يا ملك؟
الأوكسجينيتد with the oxygenated blood.
Yes,
فهيكون
exactly.
في less
حاضر يا بودي،
يعني
حاضر حالا. يبقى الاوكسجينيتد with deoxygenated blood will be mixed, so less oxygen. If they both mixed يا ملك، مش ده معناه انه less oxygen will be pumped to the body? Because the blood became mixed now, not pure oxygenated blood. You got it? Okay. Okay. So the last point to end your answer with إنه إيه يا ملك اللي will be delivered to the cell, to the tissue, to the respiring tissue. Less oxygen, sorry, to the respiring tissues, أو to the tissues. Abdurrahman, C. Could you please read the question and then less oxygen, ya Bara. Don't talk about the structure of the red blood cell. Less oxygen, not less hemoglobin. You got it? Less oxygen will be delivered to the respiring tissue because hemoglobin is found بس البايندينج between الهيموجلوبين والاكسجين هو اللي got affected. فانت هتقولي less oxygen to be delivered. طيب, could you please read the question يا ولاد? And then I asked already Abdurrahman to describe the data for aerobic and anaerobic respiration in front of you. Abdurrahman, you will say it loudly ولا هترايت في الchat box? طب يلا. اوكي. طيب تعالوا نفكر فيها قبل ما عبد الرحمن يكتبها يا ولاد. هو he will write the answer in the chat box. Okay. This figure shows how the body uses aerobic and anaerobic respiration during the first 120 seconds of vigorous physical activity. Between 0 and 20 seconds, the body also uses stored energy. And he wants you for 4 marks, and you should ask me what is the total mark of the question in order to answer, to describe the data. Okay, I can say for aerobic respiration something, and I can say, I mean 2 points for aerobic respiration, and I observed already 2 points for anaerobic respiration. Okay, so لسه عبد الرحمن is writing. خلينا نتشيك together the general point اللي هسرق بيها أول mark from الexaminer. Okay, إنه both aerobic and anaerobic increases at the start. Okay, ماشي. عبد الرحمن اداني answer، بس هي أول point اللي أنا قلتها دي تاخد بيها الفيرست مارك يا عبد الرحمن الأول. Then he wrote for the aerobic respiration, it keeps increasing till it reach a state of plateau or levels off at 100. However, anaerobic increases till it reach an optimum at 20. So he compared between aerobic and anaerobic, and anaerobic energy increases more rapidly. However, it decreases after 20. Well, this is another point. Aerobic respiration will not decrease, it remains high. يدان ما قلتش. Even في leveling off, it reach a plateau or a levels off at the high rate. It did not decrease. I think now by just the comparative data اللي we will add هناخد ال 4 marks عادي. اوكي، يبقى الفيرست مارك.
ممكن اقول ان
قولي يا
ان لما الانرجي في الانيروبيك increase بعد كده decrease، بس لما decrease didn't reach زي ما كانت في الستارت.
ماشي no problem، بس هو ده هيرفلكت ازاي على الداتا؟ ما انت you need to describe something you might use for in the explanation next. Okay, so you can say about انه after 20 what happened، وبعدين بتreach levels of between what and what. عندي هنا increase then it remains high. قلنا في الأول البوينت بتاعت إنه both aerobic و anaerobic increases at the start. الاتنين increase at the start. وقلنا كمان إنه الانيروبيك doesn't خلاص احنا كده we have covered ال 4 points. احنا بنعيد نفس الكلام.
اوكي.
اوكي طب يلا يا ملك قولي لي بقى البالانسد chemical equation for aerobic respiration.
C6
ايوه
H12 O6 plus
plus
6O2
exactly
equal 6CO2 plus
plus
6H2O plus energy.
okay يعني anyways because it is a balanced chemical equation he wants to check the equation and the balancing. اوكي يعني fine nice ان انت تقولي ال ATP او الانرجي بس يعني anyways مش هنلوز مارك لو نسيناه. During الanaerobic safe during الanaerobic respiration an oxygen that can build up state the name of the chemical that causes this oxygen that ايه chemical substance اللي produced from an anaerobic respiration in muscles. Yes exactly lactic acid. Abdurrahman answered. Okay lactic acid. Let's go to question 3. This is a growing plant والplant ده بيgrow in the dark. عايز النيم بتاع الجروث. Response shown by the plant shoot in figure 3.1. And explained how the growth of the plant shoot is controlled. I can see some answers. هو اللي بيقول phototropism he said at the beginning إنه this is a dark الplant is growing in the dark فالشوت هنا is going away from the gravity so there is a gravitropism on the shoot يا دان. بلاش نلينك دايما إنه الشوت is growing towards the light. This is a fact when we have light. But if we don't have light who is giving you a tip في الhead بتاع الquestion we have dark area the shoot will grow away from the gravity so there is a negative gravitropism response from the shoot. مش هو كده يا دان بيgrow away from the gravity بس فين اللايت to write phototropism there is no light so this is called negative gravitropism and it's negative يا ملك because it's away it's going upward against the gravity. Okay then you got it.
Yes, I got it. Thank you.
okay so please read the question well based on the question we can write the answer. Explain how the growth of plant shoot is controlled. Controlled it means it's regulated by what? Regulated by الأوكسينز اللي made في الشوت tip and diffuses or goes down في الشوت cause cell elongation. Okay? Causes cell elongation. Yeah, there's no need to mention we can say lower sides يعني يا ملك instead of the shaded area because we don't have light. And you can say a lower side or accumulates on the lower side of the stem instead of saying shaded area. ما أنا قلت بقى من الأول أنا ما عنديش light. أوكي؟ فمش هقول shaded area بس عشان نكون بنكتب صح. Let's go to the next question. I told you one day إنه they did not ask for uses of water for a while now. I expected this question in our paper, but it came now in March. Okay? يعني they did not ask or test the use of water. ماشي. Okay. برافو عليك. Okay. Some of you mentioned cooling. Okay. ان هي used for يا ولاد هو احنا بننسى ليه الفوتوسينثيسيس؟ Yes, exactly يا عبد الرحمن. الفوتوسينثيسيس should comes first. Okay. يبقى الفوتوسينثيسيس supporting tissue structure, plant cell, turgidity, used as a solvent, transport medium for minerals مثلا يا ملك. خلاص أنا I said 4 now, not only 2. Let's go to C. Percentage increase. Could you please tell me Dan readings اللي you will get بس وبعدين مش مشكلة calculation now. أنا I need بس reading. And 48.
It was 9 at
It
48.
was 9. Bravo.
And 96, it was 25.
Okay. And percentage increase يعني final. It was كام and 96 يا مالك sorry.
25.
Okay. 25. Yes. Okay. بركز معاكي بس. So احنا we set final minus original divided by original times 100. This is the formula. Okay. So 25 minus 9 divided by 9 times 100 نجيب الانسر to 2 decimal places وكده نبقى خدنا 3 marks. Please don't forget يا Dan to show him your working. Question 4 about hormones, female hormones. Okay. And he's giving you a concentration of the hormones in the cycle. He is asking you to identify hormones R and S. What's hormone R and what's hormone S? Hormone R is the estrogen, and hormone S is the luteinizing hormone. If you know يا مريم يا ملك يا عماد انه R هي الاستروجين خلاص one hormone left وهو الاس اللي بيبقى موجود او بيكوز الرابتشر اور الاوفيوليشن الايج follicle rupture or الاوفيوليشن which is LH. يلا next question. الاوفيوليشن mainly based على الجراف here from day 13 to 16. This is the time of ovulation. Okay. And the shedding of the uterine lining برضو based on the graph here from 1 to, we can say 1 to 7, you can say 1 to 6, you can say up to كمان 8 it will be accepted. Okay, arrange يعني. They will be arranged. بس يعني I will go for from 1 to 5 أو from 1 to 7. Usually يعني بنكتب numbers دي. Question C. Describe how the concentration of progesterone would change during a pregnancy. What do you think? We'll give a reason for the change. خدوا بالكم I'm telling you during the pregnancy مش during the menstrual cycle. During the pregnancy, and it is 2 marks. What will happen إنه تفتكري يا دان ولا أنا محتاج thick layer for implantation. It will increase gradually because we need to implant the embryo in the uterus.
Sir, but we're maintaining the lining, right?
Yes, well, this is this is during the pregnancy. We are maintaining the uterine lining by the secretion of the estrogen of the progesterone from the placenta.
Yes, no, that's what I
So
meant. In
yeah. So the
need to just
concentration will
interpret it.
increase. Then you can mention and will be constant during the pregnancy week to maintain the uterine lining. يبقى هتقولي increase الأول وبعدين ممكن تقولي ان هي it will be the same او because we need. انا لو مكانك بصراحة مش هكتب ان هي it will be the same. انا I will write because he wants a reason, so I will write it will increase in order to maintain the uterine lining وكده هبقى واخد ال 2 marks.
Okay.
Okay. Question D. State the sites of production of progesterone. يا سلام. During the menstrual cycle و during the pregnancy. During the menstrual cycle الcarpus luteum is اللي هو الساك أو الfollicle part هي اللي بتproduce البروجسترون. And during the pregnancy what البلاسينتا, and that's what we mentioned. Okay. By the way, if you wrote يا عبد الرحمن الأوفري لا, it will not be right for the pregnancy. If you wrote الcarpus luteum, no, it will be incorrect برضو. فانت during the pregnancy لازم هو بيتست you في انك عارف ان البلاسينتا هي اللي بتسكريت البروجسترون during the pregnancy. Describe the role of FSH in the menstrual cycle. FSH, the first hormone from the name, بتستيميوليتس ايه؟ بتستيميوليتس maturation of the follicle. كمان بتستيميوليت releasing of estrogen او estrogen secretion from the ovary. فاكرين السايكل اللي احنا عملناها في الكلاس starts from FSH then estrogen اللي increase وبيبيلد اب اليوترين لايننج وبيريتش البيك. ساعتها بنستوب FSH وبنسكريت LH for ovulation or rupture of the follicle. And finally الcorpus luteum اللي بتسكريت البروجسترون. Let's go for question 5. This is a monkey flower plant. هو عايز 2 pieces of evidence visible. عشان خاطري the word visible is important. You could take the 2 marks or lose the 2 marks. Okay? That show that monkey flower plants are pollinated by insects. Yes, Salam. I have large petals. Yes, exactly, then. Large petals. I have what else? I can't say nectar guide here, but I can say spotted pattern. With this spotted pattern برضو بتattract الانسكتس يا ولاد. Okay. I can say إنه الspotted patterns دي بتattract الانسكتس. Okay. هو أنا مش قادر أشوفها يا دان إن هي الفيلامنتس والستايل inside the flower. I can see well بالنسبة لي it's not visible. Okay.
So if I say they're not protruding out of the flower, would that be correct?
يعني I will not go for this here then, honestly.
Okay,
Because
so
I
what's
need
the
to.
second point?
That's what we said. Large petals with spotted pattern. قدامي اهي all of these spotted pattern attract the insect as well.
Oh, okay.
يعني it's like النكتر guide اللي كنا بنكتبها, بس here we have already spots instead of a nectar guide.
Okay.
Okay. Or we can say the spotted pattern D used as a nectar guide to attract the insects. Okay. يلا بينا. Double I. The monkey flower reproduce sexually, and I want you to describe the advantage and disadvantage of sexual reproduction for monkey flower plants. What's the advantage and disadvantage? Please take a look on those tables في السexual والasexual. Okay. الادفانتج والdisadvantages of السexual. أول ما بقول advantage للsexual advantage, we should mention variation, increases variation, increases ability. When we have more varieties of plants, this means that we have more ability to adapt, to change. When we have more variation, we have more plants could be resistant to some diseases. All of these could be the advantage. But what about the disadvantage? It requires two parents. It takes longer time. It requires pollinators. All of these are disadvantage. Okay, let's go to B. Type. This is a comparison between a leaf area of 5 monkey flower plants growing in Europe with 5 monkey plants growing in North America, and he's giving you the plant number and the mean of the leaf area per centimeter square for both, and he wants you to calculate the mean. How could we calculate the mean, يا ولاد? And add divided by 5. and add all together divided by 5 هنقدر نجيب المين. وبليز write the unit which is centimeter cube. 1 mark will be awarded for the value and 1 mark will be awarded for the unit. State a conclusion of the results from the table. I can say in the plant اللي في Europe has what? Greater leaf area than الموجودين في North America. Overall نقدر نعرف ده من المين. المين by the way here هيكون 301. فdefinitely هنقدر نعرف ده من المين. يلا. I will give you like 2 minutes trying to answer this question. Explain how the monkey flower plants have developed a larger leaf area over time. Is it natural selection idea? Is it natural selection or not يا ملك يا عماد? Is it natural selection or not يا براء? Okay. When he mentioned have developed and he's talking about something really developed in the environment or in specific or particular habitat, so it is a natural selection idea. Okay, we should explain بقى natural selection how we got genetic variation and how some living organism will struggle to survive, some living organism or some monkey flower plants has a greater chance of survival. large leaf area هي اللي هتبقى عملت better adaptation, reproduce, pass the genetic material to the offspring over many generations. خلاص, I got the idea of the 5 marks now. Question 6. Yes, Salam. Question 6, he's asking about ازاي الفيتوبلانكتون وهو بيقول لي ان الفيتوبلانكتون فيها chlorophyll. So it is an extremely easy question. You should talk about in here producers using the photosynthesis process, so it will use carbon dioxide and requires light energy. Suggest the role of zooplankton in the ecosystem. The zooplankton considered as what? هم eating krill feces, so they are feeding on الkrill feces, so they considered as decomposers. Two processes add carbon dioxide to the atmosphere. The presentation. What are these? Respiration and combustion. Exactly. Respiration and combustion. Nice. Nice answer by chat box. Thank you. Describe the effect of the environment on additional carbon dioxide in the atmosphere. You need to talk about carbon dioxide is a greenhouse gas. This will increase the temperature أو global warming. This will cause a greenhouse effect. This will change the climate. The sea level increases. It might cause habitat destruction. برضو يا ملك, I'm giving you more than 3 marks. You should know all of this. Question B. Figure 6.2 shows the pyramid of energy for a food chain in an ocean ecosystem. He wants you to label the trophic level containing herbivore. The herbivore will be on what level? Herbivore, it means living organisms feed on plants. Explain. لا ده easy question بقى وجي مليون مرة خلاص. law of 10%. Okay. actual energy transfer amount of energy will be lost 90%, such as heat from respiration, non-edible part, feces, excretion, reproduction, movement. So I think this is an easy question. Two advantages of using a pyramid of energy rather than a pyramid of biomass to represent a food chain. What is the advantage of this? A pyramid of energy gives you the actual energy transfer. Okay? And all of the living organisms will be in the pyramid of energy. There is no need to kill an organism in order to know the biomass بتاعته. Okay? But all of these are the answers. What's meant by the term sustainable resources? Resources produced as rapidly as it is used, so it does not run out. Excuse my handwriting بقى. But you know my handwriting well. Let's go to last question in this paper. Fishing can be managed to promote the conservation of fish stocks. Explain one way that fishing can be managed to conserve fish stock. In the class, I mentioned, we could increase awareness or encourages people not to overfish. We could prevent overfishing by having quotas, by monitoring the rules of overfishing, and we can also say in we will use diameter, big size diameter for the net. Okay, and we can also use we need to allow fish to breed in some closed seasons without overfishing. Okay, now we have covered this paper. Both exams was really nice. برافو عليك يا عبد الرحمن. Thank you so much for you all for your participation. I know it's not that easy by the end of the day to take a biology class, but thank you so much all for your participation. I encourage you next week to attend the online sessions as well. We will answer a lot, and if you have any part that you want me to clarify and explain, please let me know before the session. Thank you so much, حبايبي. Bye.
باي مستر.
Bye.